My name is Jason Metcalf, and I'm a G3 in the Department of Biological Sciences. And today I'm going to talk about an evolutionary perspective on new sources of antibiotics. So I wanted to draw the lights a little bit so we can see things. I wanted to start with what I think is a familiar image to everyone in the room. This is the Parthenon in Centennial Park, just down the road, on a beautiful sunny summer day. I think anybody walking through this park or looking at this image thinks it's a nice place to go for a stroll on a nice afternoon. But if we had superhuman vision that could zoom in and look at the same scene on a microscopic level, it'd be a completely different world. We'd see that the water is just churning with all kinds of microorganisms. We've got cyanobacteria and protists and algae, all kinds of things. The trees basically have smaller versions of themselves and growing on their leaves and growing on their roots. The soil is churning with all kinds of decomposing bacteria and fungi. Even the humans running in this park are by no means clean organisms. It's been estimated that for every cell that we have in our body, there are 10 microbial cells, either growing in our gut or on our skin. So in some ways, we're more microbe than man. And if we zoom in even a little bit further and start looking at the viral realm, things get even weirder. We see these strange creatures that, well, maybe not creatures, but conglomerations of protein and nucleic acid moving back between cells, killing cells, bursting out and sort of spreading all over the place. When you look at things at this level for long enough, you start to realize that Earth is less a nice, clean, pristine place of which we are the master, and it's more like we're a bag of microbes wandering through a world dominated by them. Now, before you start running off to the bathroom to start scrubbing, realize that this is not necessarily a bad thing. And the reason I say that is because for every big, bad, scary microbe that may cause us harm as a pathogen to us, there are a million more that are frankly on our side. All the millions of commensal bacteria that live in our gut and our mucosal membranes on our skin are protective. They prevent these harmful pathogens from colonizing us, either by just taking up resources and occupying a niche, or by actively secreting compounds that can inhibit these bacteria called bacteriocins. Now, thinking about the world with all these trillions of microbes in it, we have to add another layer of complexity to it because all of these microbes are interacting. Microbe A might be competing for resources, the exact same resources that microbe B is. Another might be secreting a molecule that kills a different species. Many of these microbes are secreting compounds that let other members of the same species know the concentration of that species so they can coordinate their efforts and produce products at a specified time. Another might be infected with a bacteriophage that's going to infect all its neighbors, and it goes on and on and on and on. And before long, you realize that the microbial interactions in just a handful of soil is so mind-bogglingly complex that we have no hope of ever comprehending it fully. That being said, some of these interactions I think are very important and merit further study. One of the reasons I say that is because every microbe in this gamesh has friends and has enemies. And many of these enemies are actively seeking out to kill a pathogen that we might be interested in killing. And also, don't forget that it's not just microbes that are doing battle here. We are not the only species on this planet that have enemies in the microbial world. Every eukaryotic species that's ever existed has had to contend with these microbes, from plants to fish to dogs and cats and everything. They all have their innate immune system, and they also have a repertoire of innate immunity antimicrobial compounds, which is the subject of my research. Now, before we simplify things, we're going to add one final layer of complexity to all this, because you have to realize this is not taking place in a static bubble. It's happening over evolutionary timescales. And we as humans have known about a pathogen like Staph aureus for maybe 100 years, and we've tried to prevent blood poisoning for maybe a few thousand. But as long as prokaryotes have existed, they have been warring with each other for billions of years, and evolution has had a huge leg up on our development of antimicrobial weapons. So I really think taking an evolutionary perspective to development of antimicrobial compounds can teach us a lot. And one of the most important things that I think it can teach us is this idea: the enemy of my enemy is my friend. Every microbe that we call an enemy has its own enemies, and we need to make them their friend, and make them our friends. Maybe using their own antimicrobial weapons as a therapeutic. So with that idea in mind, I'd like to introduce you to one of my favorite microbes called Wolbachia. Wolbachia is a genus of obligate intracellular bacteria. That's probably one of the most widespread infections known to man, and it's a favorite model organism of the Bordenstein lab. It infects about half of all arthropods in the entire world, so it's really an enormous infection. And it does some really cool manipulations of its host. In one particular lifestyle, Wolbachia is a reproductive parasite. It actually manipulates its host species so that it can further its passage down through the generations. Since it's only passed from mother to offspring, Wolbachia has evolved sort of a man-hating attitude and really serves to benefit the females of a species at the expense of the males. In some strains, Wolbachia will outright kill most of the males in a population so that the females have a better chance. They can turn genetic males into females, and they can even turn an entire species parthenogenic and just remove males from the equation entirely. So there's some really amazing manipulations that it does, and we're just starting to understand how it does these things. The other lifestyle that Wolbachia has is sort of the opposite of parasitism. It's obligate mutualism. In the case of bedbugs and in filarial nematodes, the two actually need each other to survive. So if you kill the Wolbachia, you can also kill the bedbugs. The Wolbachia provides certain vitamins that bedbugs can't make themselves, and so there's mutualism going on there. Adding to this story is that it's not just a two-way street. There are multiple levels of parasitism and something we call hyperparasitism, parasites of parasites. Wolbachia in the lab we often grow infecting Nasonia wasps, which themselves are parasites. They lay their eggs in developing blowflies. It stings them, paralyzing them. The eggs hatch and eat the blowfly alive from the inside out. Blowflies themselves are parasites as well. They lay their eggs into dead and dying animals. So you can see how this is hyperparasitism with multiple levels of parasites. There's also multiple levels of mutualism going on here. In the case of the filarial nematodes I mentioned, Wolbachia and the nematodes need each other to live. And these nematodes can cause some pretty severe diseases in humans. In sub-Saharan Africa, diseases like lymphatic filariasis or elephantiasis and river blindness are caused by these worms. Since the Wolbachia, or the worms need the Wolbachia to survive, you can imagine there's been considerable interest in generating anti-Wolbachia compounds that can kill the Wolbachia so the worm also dies and you can cure the infection. In the Bordenstein lab, our idea has been to take this hyperparasitism one step further, because Wolbachia itself is parasitized by something called phage Wo, which is a bacteriophage, a virus that affects bacteria. It's a temperate phage, which means it spends part of its life cycle integrated into the genome of Wolbachia, and then under certain circumstances it becomes lytic, bursts out, kills the cell. And so part of my thesis has been trying to identify what molecules or proteins does phage Wo use to kill Wolbachia, in hopes that eventually we might use it as a therapeutic against these filarial nematodes. It's sort of taking this enemy of my enemy is my friend paradigm one step further, and then we're taking a protein from a virus to kill a bacteria so the worm dies and you cure the infection in the human. It's sort of a multi-level system that has some pitfalls, but I think it's a really cool idea. And so that's how my thesis started out. Find anti-Wolbachia proteins that phage Wo has in its genome. So when we first started looking for what genes might be involved here, we found several that were pretty interesting, but the one that I want to talk about today is called a lysozyme. Now, lysozymes are enzymes that degrade peptidoglycan, peptidoglycan being the sort of net-like mesh that covers around the inner membrane of pretty much every bacterial species. If you break open the peptidoglycan layer, like lysozyme might do, you can kill the cell, burst open, it spills its guts, and it's dead. Bacteriophages have long used this at the end of their life cycle to burst open the cell and release their progeny. This is shown here in a video where E. coli is infected with a virus called coliphage T4. And you can see that within seconds of the lysozyme getting access to the cell wall, the cells burst open. Those are, of course, bacterial screams you're hearing. Don't be alarmed. So this seems like a very likely candidate for phage Wo being used to kill Wolbachia if other phages are using the same enzyme to lyse their hosts. So being an evolutionary lab, the first thing we wanted to do was sort of get a look at the evolution of this gene and where it may have came from. To do this, we blasted the gene against the NCBI database to look for any homologs in other species, trying to figure out where this gene might have came from. We found quite a few homologs in other bacteriophages. That's to be expected, of course. We also found it in a lot of bacterial species, which at first doesn't make sense, but then you realize that all bacteria have similar enzymes themselves. They use them in a regulated fashion, of course, to split whenever they split their peptidoglycan layer whenever they're dividing, and to do some remodeling of their cell wall. So it doesn't wipe them out, but you use it very carefully. So we found it in expected places, and then we also found it in a few places that we did not see coming. One was an insect, a pea aphid. Another was several different species of fungi. We found it in a plant called Selaginella that's a spike moss, lives in China. And we found the same gene in an archaea called Acidulae profundum boonii, which lives in deep ocean hydrothermal vents all around the world. So these are sort of weird places to find this gene. Archaea, the third domain of life, is just as different from bacteria as we are. So it's a little weird that they have this enzyme, considering that archaea don't have peptidoglycan. None of these species in this here at all have peptidoglycan cell walls. So it begs the question, why do they have this enzyme that degrades peptidoglycan if they don't even have it? Our hypothesis has been that at some point in evolutionary past, each of these species basically stole the gene from a bacteria and is using it now as an antimicrobial weapon. One interesting corollary of this hypothesis is that bacteriophages, their lysozymes, are usually very tailored to attack only the host species that the phage infects. It doesn't make a lot of sense for a phage to burst open its host and then kill every bacteria in the neighborhood. It's evolved just to attack its species of interest. However, you can imagine by moving this gene into an entirely different species, into an archaea or a eukaryote, there's going to be very different evolutionary pressures. You might want to fight off multiple bacterial infections instead of just this one species. So our hypothesis has been that these species have stolen this gene and have evolved a broader range of activity of the lysozyme. And so that was our hypothesis moving forward. For most of the talk, I'm going to focus on the archaea lysozyme, partly because it's a really cool place to find this gene, and also because we've never identified any antibacterial molecules from archaea before. We know they can kill bacteria, but we have no idea how. And so this would be interesting to identify. I'm going to be comparing the lysozyme activity in the archaea to a lysozyme from a bacteria and a phage. So the first step was just to make sure we're not crazy here, that there really is a lysozyme gene in this archaea species. It's not an artifact from sequencing or assembling the genome in the database or something. So we worked with a collaborator to actually get samples from hydrothermal vents all over the world. And in the green stars are all places where we found this gene, not just there, but integrated into the archaea genome. We found it in 4 different places in the mid-Atlantic ridge and in the western Pacific. So the gene really is there out in the wild. This begs the question, why is it there and where did it come from? So in order to try to address that, I took the sequences of all of these different wild isolates of the gene, added it to the NCBI data that we pulled from databanks, and created an evolutionary phylogeny. This is the only tree I'm going to show you, and there's a lot of information here, but the only thing you need to know is that A, it's color-coded. In black are all the bacterial species. You can see that makes up most of them in this tree. And then in red are the archaea. And you notice that they all group together, all the archaea species, all the strains are all together, and they all come from a branch in a clade that's dominated by bacteria, in fact, from the phylum Firmicutes. So what this is suggestive of is something called horizontal gene transfer, where a gene, probably from the ancestor of these Firmicutes, was transferred from that ancestor to an ancestor of the Boonii. That was then propagated down to all its offspring through the strains as these branched out over evolutionary time. Okay, so the next step was because we know that horizontal gene transfer happens pretty regularly in bacteria. They're swapping genes all the time. But for a gene to move from a bacteria to an archaea or to a eukaryote, that's pretty rare. And when it does happen, usually the gene gets lost because we don't usually have a need for any bacterial genes. And they may not be regulated well, and when it gets swapped into our genome, deleterious mutations accumulate and the gene gets lost pretty rapidly. That's called pseudogenization, and we wanted to check for that in our gene of interest. So what I've done here is something sort of similar to some stuff that Pedro talked about last week as far as protein modeling. What I've done is taken the sequence from several of these lysozymes and then used a program that blasts them against databases that have all the solved crystal structures of every protein that we've ever solved. It then tries to build a model based on similarities between the protein of interest and those proteins that have already been solved. And so I'm showing you models for a bacteriophage lysozyme, the one from archaea, as well as the one from the plant. And you can see they all have very similar structures. They all have this same beta barrel motif. And in purple and red, I've highlighted the residues that are most highly conserved across that entire phylogeny. They're all in this central beta barrel, which is where the active site is. The residues in red are actually the catalytic residues, and they are universally conserved across all the lysozymes in that phylogeny. So it really looks like this enzyme works, even in these new species where it first doesn't seem like it should be there. But of course, we don't want to just predict that it works. We want to show that it does. So in order to test these lysozymes' antimicrobial activity, I took the gene from the archaea, and as well as the bacteriophage and the bacteria that was most closely related to it in that phylogeny I just showed you. I cloned each of those genes and then had E. coli produce the proteins for me. I then purified them, checked them out on gels, and then added them to different bacterial species to see what kills what. So first I just want to show you that I was able to get pure enzyme. I have the bacteria and the phage and the archaea lysozyme, and the first column in each section is just a crude lysate of the E. coli. Burst open the cells, what proteins are there. And then you can see that I purified the protein and removed all the rest of the gunk. So I've got pure elutions of each of these proteins, which I can now add to any bacterial species I want and see whether or not they die. So this is one of the coolest figures I think I've done in grad school, which I'm really excited about, is showing the antibacterial activity of these lysozymes. In each row is a different species of bacteria, and in each column is a different lysozyme. The first two rows are the bacteria and the phage, and you can see that all these bacteria live, which makes sense because bacteria don't want to kill themselves with their own enzyme, and bacteriophages usually attack just the strain from which that lysozyme came from, which is not represented in these species. However, you look at the archaea, and it's wiping out several of these species and inhibiting the fifth, which is really exciting because this really is an active antimicrobial molecule in an archaea, which has never been discovered before. The final column is just a control where I've produced another protein, cyan fluorescent protein, with the same methods that I've done all the other lysozymes, but it doesn't kill. So this is pretty exciting. Another cool thing about this data is that I've organized the bacterial species in order of increasing evolutionary distance from what we think was the donor of the lysozyme way back in evolutionary history, that Firmicutes. And all these species that it's killing happen to be Firmicutes. So it really looks like the Ebunii stole this gene from a Firmicutes and now is using it to kill Firmicutes in the environment. Another cool thing about this is that if you look at the hydrothermal vent microbiome and actually examine what species are down there, you can plot the number of bacteria that are Firmicutes against the number of archaea that are Ebunii. This is just a schematic representation of this, but we notice that whenever you get a lot of Ebunii in a particular vent, the Firmicutes drop way off. In fact, the samples where we have the most Ebunii have no Firmicutes whatsoever. So it's just correlative, of course, but it really looks like Ebunii is using this as an antibacterial weapon against Firmicutes. So far, we've sort of taken an in silico approach to examining the evolution and structure of these proteins, in vitro approach to examining its antimicrobial properties. The only thing left to do is sort of try it in vivo. Now, you may have noticed Nashville doesn't have a lot of deep oceans or hydrothermal vents. So in order to work with this bug, I had to leave Nashville. I went to Portland State University to the Center for Life in Extreme Environments. There I worked with Dr. Anna Louise Reisenbach and her postdoc Kristen Berleya. Dr. Reisenbach was actually the first person to go down with the submersible subs and take these samples from the bottom of the ocean, bring them back in lab and culture them. So there's really no other place in the world that I could go to work with this bug. And of course, we had the moral support of the lab pet, Dr. Reisenbach's dog. So the idea behind the experiments I was doing in the Reisenbach lab were to culture Ebunii either alone or to add a bacterial competitor. The idea being that if Ebunii is really using this molecule as an antibacterial weapon, it either can express it all the time, which wouldn't be very efficient, or it could crank it up expression whenever you add a bacteria to the mix. So that was the idea, culture Ebunii either alone or plus a bacterium and see what happens. Now, as you can imagine, the culture of these organisms is not typical. They're obligate anaerobes, which means there can't be any oxygen at all. You have to bubble through carbon dioxide and hydrogen gas. And then all this cultures get sealed in these anaerobic culture tubes. They're methanogens, so each tube gets a spoonful of elemental sulfur, which you can imagine smells wonderful. And if you remember from the name, it's a sigilly profundum bunii. It likes acid. This is growing at pH 3.5. They're growing at hydrothermal vent temperatures, so like 70 degrees centigrade. we're basically culturing these bugs in hot sulfuric acid without oxygen. It's very unusual culture conditions, but it was a lot of fun to learn how to do that. Now, I just brought these samples back to Vanderbilt to extract RNA, and I can tell you so far that it looks like the gene is expressed at a basal level in cultures alone, and hopefully in the next couple of days I'll be able to tell you whether or not the gene is upregulated in the presence of bacterial competitors. So stay tuned for that. So that's where the project stands so far, and I just want to wrap up with a couple of big picture take-homes that I hope you got from this. One is that we live in a microbial world. Microbes are in and around us everywhere, and the interactions they have are probably pretty important, and I think there's a lot we can learn from them. Secondly is to remember that paradigm I told you, that the enemy of my enemy is my friend. Every pathogen that affects us hurts other organisms too, and they already have their own antimicrobial weapons that we might be able to use ourselves. Thirdly, I think evolution is a really important aspect to take in pretty much any biological problem you look at. It's been said that nothing in biology makes sense except in the context of evolution, and I think that's really important. So I encourage you all, if you have the opportunity, to take a biological or a evolutionary approach to any of your projects, to do so, take that opportunity. And finally, I'd like to say, just don't be afraid to follow the science wherever it leads you. I started working with insects and flies in the lab, ended up with archaea at the bottom of the ocean, and Portland State University with someone I never worked with before. So don't be afraid to follow the science, work with new organisms, work with new people. I think it can be very rewarding. And with that, I'd just like to thank the Bordenstein Lab and the Reisenbach Lab for all their support, and you guys for listening, and I'll take any questions. Yes?
So looking forward, how do you envision using lysozyme as an antimicrobial?
So we're not entirely sure how much or what concentration we'd have to give it to work on like a human infection. But even if it is really high, we could use it as a topical thing for sure. There's been some studies that have been trying to use it in eliminating carriage of like MRSA in the Nearies, things like that. There are a lot of options there. Yes.
When you're saying that the lysozymes are similar in sequence, like when you're looking at NCBI blasting it, like what's your cutoff? Like how similar are they?
So when I first got all those sequences to make that phylogeny, I just did a blast search with the Wolbachia phage lysozyme as the query, and anything that had like a 10 to the negative 10th E value or higher, I threw in that phylogeny. So they're, in some cases, they're like 50% identical between, if you look at any particular couple of lysozymes.
Yeah, because I wonder like if like their function is truly the same in each of these organisms.
Yeah.
And I don't know if there's a good way to test that, or if even an organism can survive without that gene, if it's actually necessary.
Yeah. Well, if you look through the NCBI database, of which there are something like 400 or several hundred sequenced plants and 400 sequenced archaea, none of them other ones have this gene. So I don't think it's going to be necessary for every organism. I think that some of these organisms got lucky, picked up the gene, and now it's been selectively, it's advantageous for them, and so they've held on to it. Interestingly, A. boonii is one of the most successful archaea in hydrothermal vents. I have to wonder why that is. Maybe this is one of the reasons it has a repertoire of armaments to kill bacterial competitors. Yep.
Did you say one was not a pseudogene in other species?
In all the species I showed you in the phylogeny, it was not a pseudogene. It looked like the active sites were still there.
But in BLAST, it picks up all pseudogenes, right?
Anything that has a similar sequence, and you just have to look at it to see if you think it's a pseudogene or not.
Okay.
Yeah.
So there were other species that had pseudogenes.
Well, I mean, if you go far enough down the blast search, you will always pick up junk at some point. But I don't think any of this is junk. Interestingly, the plant does have like 4 copies of this gene in it, which is not that uncommon for plants to do because they often duplicate their genomes. And a couple of those other copies look like they may be pseudogenized, just because the plant doesn't need all of them. They probably only wanted the one. Yep.
Is there evidence of resistance developing in bacteria populations to this kind of enzymatic or antimicrobial approach, and how would that affect the use of this enzyme as a clinical outcome?
It's a good point. There are some species that have natural resistances, and that may be due to the structure of their peptidoglycan. But the thing about beta-glucan is it's absolutely required for virtually every bacterial species. And the lysozyme attacks a conserved linkage there that just the bacteria can't do without. So they've been experiments where they've taken lysozymes from other families and put them at very low concentrations and let the passage bacteria, 10 generations or more, 40 generations in one experiment, and they never developed resistance. They always still were susceptible to it. I'm not saying it won't happen, it can't happen eventually, but it's probably going to be harder than evolving resistant to a small molecule inhibitor. Yes?
So for the last or the current experiment that you're working on where you have the archaea in culture and then you're adding in the bacteria,
Uh-huh.
how does one control for expression in the bacteria for this thing that it uses itself to remodel its beta-glucan wall so that you don't, just because you added some bacteria to it, confuse that for an increase in expression for the archaea?
So the differences between the lysozymes in the species that I'm adding and the lysozyme in the archaea are enough that the primers that I've chosen should only amplify the archaea. But I'll have controls with just to show that it doesn't amplify those other species. Yeah, that's a good question. Yes?
Can you talk a little bit about fermentating IFA? It looks like so they're gut microbes and they're in system events.
Uh-huh. There are a lot of different firmicutes that live all over the place. Ones that you've probably heard of, Bacillus cereus causes food poisoning. Bacillus anthracis causes anthrax. There are species that grow in soil. There are species that grow in the ocean, all over the place. Yeah. A pretty diverse phylum. I mean, most bacterial phyla are very diverse, and you can find them all over the place. Yeah. Mm-hmm.
So would this lysozyme work against those?
Theoretically. I would hope so. Yeah, I haven't tried it yet. We only have BSL-1 in most of our lab, so I have a BSL-2 room set up that I eventually will probably try some of these. Not anthrax, obviously, but maybe be serious. It'd be cool to try anthrax. We need a collaborator for that.
Will you try to do that?
I haven't actually planned on it, but I'm thinking about it. I think it'd be cool. I mean, I only recently just showed that it was able to kill these species, so I haven't gone too far yet.
It'd be really interesting for anthrax because it's topical.
Yeah.
A lot of the
Yeah,
infections are-
absolutely, which would be a really easy application of an enzyme. Yeah, it's a really good idea. Yep.
How does this lysozyme compare to human lysozyme?
So there are actually 5, or some people think 6 families of lysozymes, and most of them seem to have all evolved independently into 6 different families. So the lysozyme that you find in us and chicken egg white lysozyme is significantly different. There's no sequence homology whatsoever. There are like 3 families that the structures overall look very similar, but there's no sequence homology. So it looks like evolution came upon the same idea at least 5 or 6 different times, and 3 or 4 of those times ended up with the same kind of protein, and a couple times didn't. This one's very different than any of the other lysozymes.
So how does human lysozyme then compare in terms of antimicrobial efficiency compared to this lysozyme that you're looking at?
It's a good question, and I haven't actually done any experiments to see the breadth of activity of human lysozyme. I know people have done it, but it's been like experiments since the 40s that anybody's ever checked that. So I'd have to go back and look. A control that I didn't show you just for simplicity was using chicken egg white lysozyme, and it seems to kill similar species, but they don't overlap always. Some species that the chicken egg white lysozyme kills, this one doesn't kill, and vice versa. So they're definitely not overlapping entirely in their spectrum. Anything else? Yes.
A very tangential question, but I'm curious about the bed bug thing. So if people try to kill Wolbachia,
Yeah, so
kill bed bugs, I know they're really hard to get rid of.
they are really hard to get rid of. And so there are a couple things about that. There's a group in Japan who actually presented the work showing that Wolbachia was necessary at a conference I was at a few years ago. And they're very interested in developing that. The only problem is, especially in the United States, the NIH does not consider bed bugs a pathogen or a vector because they don't transmit any diseases to us. They're just really freaking annoying. So they don't fund research on bed bugs. And so there's only a few labs that have the resources to investigate bed bugs. Maybe Orkin or something can fund that. But yeah, the NIH is not that interested in it. Anything else?
Do you get to go to any of those places that are starting on that?
No, I didn't. No. Maybe someday if this collaboration moves forward. Yeah. Thanks, guys.